Current Biology, Genomic Analyses of Hair from Ludwig van Beethoven, by Begg et al., 2023. Current Biology 33, 1431 to 1447. April 24, 2023, The Authors. Published by Elsevier. Incorporated. Summary. Ludwig van Beethoven, 1770 to 1827, remains among the most influential and popular classical music composers. Health problems significantly impacted his career as a composer and pianist, including progressive hearing loss, recurring gastrointestinal complaints, and liver disease. In 1702, Beethoven requested that following his death, his disease be described and made public. Medical biographers have since proposed numerous hypotheses, including many substantially heritable conditions. Here, we attempt a genomic analysis of Beethoven in order to elucidate potential underlying genetic and infectious causes of his illnesses. We incorporated improvements in ancient DNA methods into existing protocols for ancient hair samples, enabling the sequencing of high-coverage genomes from small quantities of historical hair. We analyzed 8 independently sourced locks of hair attributed to Beethoven, 5 of which originated from a single European male. We deemed these matching samples to be almost certainly authentic and sequenced Beethoven's genome to 24-fold genomic coverage. Although we could not identify a genetic explanation for Beethoven's hearing disorder or gastrointestinal problems, we found that Beethoven had a genetic predisposition for liver disease. Metagenomic analyses revealed furthermore that Beethoven had a hepatitis B infection during at least the months prior to his death. Together with the genetic predisposition and his broadly accepted alcohol consumption, these present plausible explanations for Beethoven's severe liver disease, which culminated in his death. Unexpectedly, an analysis of Y chromosomes sequenced from 5 living members of the Van Beethoven patrilineage revealed the occurrence of an extra-pair-paternity event in Ludwig van Beethoven's patrilineal ancestry. Future directions. Genomic sequence data from authenticated locks of Beethoven's hair provide Beethoven studies with a novel primary source, already revealing several significant findings relating to Beethoven's health and genealogy, including substantial heritable risk for liver disease, infection with HBV, and EPP. This dataset additionally permits numerous future lines of scientific inquiry. This initial series of 5 hair samples, spanning approximately the last 7 years of Beethoven's life, is hoped to be expanded through the authentication testing of additional independent locks of hair, and enables future testing for infections, informative biomarkers, and exposures to environmental causes of or contributors to disease. The further development of bioinformatics methods for risk stratification and continued progress in medical genetic research will allow more precise assessments, both for Beethoven's disease risk and for the genetic inference of additional phenotypes of interest. Increases in the size of consumer genetics databases, as well as the testing of additional hypothesized relatives, both living and deceased, will lend further clarity to our understanding of Beethoven's genetic genealogy. This study illustrates the contribution and further potential of genomic data as a novel primary source in historical biography,